I'd like to, of course, thank the APA and the Hirshberg Foundation for giving us the opportunity to present our work. As you heard, Dr. Eibel is unable to attend and gave me the opportunity to speak in his place. I'm happy to be here. And I've had the pleasure of working with him for about the last year and continuing this project related to how a high-fat diet and obesity relates to the progression of pancreatic cancer. So as you know, obesity is quite an epidemic in the United States. And over the last 20 years, you can see that really the trend is not abating. This is data from the CDC from 1990 to 2010, showing you in a color-coded format that in about 1990, only maybe 10 to 14% of the American population were obese. And over the next 10, 20 years, you can see this color change is quite drastic. Now, around 2010, 25 to 29% of Americans are obese, so it's a real problem, and it doesn't seem to be abating anytime soon. So how does this relate to pancreatic cancer risk? Well, there have been a couple studies in the last few years that show us that obesity does indeed increase pancreatic cancer risk. These epidemiological studies are really quite striking in that Genkiger et al. In 2011 showed that if you have a BMI at baseline of about greater than 30 versus people with a 20 to 23 range BMI, your increase in pancreatic cancer risk is you have an increased risk of pancreatic cancer. And then in addition, if you have a BMI of over 25 in early adulthood, you too have an increased risk of pancreatic cancer. And there are also studies that show that if you, that people with cancer and pancreatic cancer specifically, that if they have an increased BMI, that mortality is increased. So this is great, but how do you what we want to know is how the adipose tissue really changes in obesity. So in a lean individual, adipose tissue has a number of different components. Adipocytes containing, of course, adipocytes, as well as a stromal-vascular fraction. The adipocytes are secreting leptin and high levels of adiponectin, and the stromal-vascular fraction contains inflammatory cells, but in a lean individual, not many, and there's not much in the way of cytokine expression. This changes in the obese state in that the adipocytes grow in size and number. There's a shift in the way adipokines are expressed. There's an increase in leptin and a decrease in adiponectin. And this stromal vascular fraction increases. More inflammatory cells, there's higher expression of cytokines. So if we understand what happens to adipose tissue in the process of obesity, we don't really understand how this actually links to cancer progression. We know that this happens, but this area here where how it's linked is pretty unclear. So as I mentioned previously, with obesity, you have inflammation, you have increased lipids, you have increased IGF signaling, insulin signaling, and changes in adipokines. And then these are all linked to increased cancer progression, but we don't know how. So we've been trying to figure out how this relates specifically to pancreatic cancer progression, because this is our interest. So as we've been trying to figure out how obesity and a high-fat diet relate to pancreatic cancer progression, we've been using a mouse model that many people have talked about in the last few days. It's the KRAS model, where you have slowly progressing pancreatic neoplasia and then low penetrance of pancreatic cancer. What we wanted to do then is to take this mouse model and create a model that's similar to human model of obesity. So using wild type and our KRAS mutant mice, we put them on either a control diet or a high-fat diet. So this high-fat diet is about 40% fat in the form of corn oil. We let these mice have free access to food and water over this time. We let them go for about 14 weeks. And then at harvest, we first imaged them. I'll talk to you a little bit later about the imaging we did. And we then basically harvested organs and looked molecularly and histologically at what happened. So you can see that on a high-fat diet, which is not surprising, these mice, of course, gained weight. They also had multiple metabolic disturbances, including changes in insulin level, glucose, IGF-1, and leptin, giving us a good mouse model for obesity. And we were interested when we took a look at the pancreas histology to find that in our normal mice, or sorry, in our KRAS mice on the control diet, they had the typical level of PanIN-1A histology, very little higher grade dysplasia and no progression to cancer. The mice on a high-fat diet, however, were less likely to have normal histology and had a greater percentage of PanIN-1A lesions, and then even a significant increase in PanIN-1B, PanIN-2, and PanIN-3 lesions. So we wanted to characterize this a little bit better and see exactly what it was that was also related to what other processes could potentially be adding to this progression. So we looked in terms of inflammation in the tissue. When we looked at the pancreas again, you could see that the wild type on a control, or sorry, our KRAS mice on a control diet versus our KRAS mice on a high-fat diet, there was an increase in acinar cell loss, an increase in inflammatory cells, an increase in stromal fibrosis, and an overall increase in pancreatitis index. That's specifically in our KRAS mice. We also saw this mirrored in our wild-type mice. And then because we saw such intense inflammatory infiltrate, we wanted to better characterize exactly what types of cells were there. So our initial stainings were to look for B cells, T cells, and macrophages. There's really not a strong presence of B cells in the pancreatic of these mice. However, there's a pretty strong infiltrate of T cells and macrophages. And then because there was such a strong infiltrate, we wanted to look a little bit more on the molecular level. So we used a multiplex assay to get an idea of the type of cytokine expression changes that we saw. This is just some of our data, and it's probably hard to read from the back, but you can see that there are changes. In gray and in black are KRAS mice, and in white and light gray are wild-type mice. You can see there's some extreme changes just with the high-fat diet, as well as between the KRAS and our wild-type mice. More specifically, I'll just talk to you about a few that we looked at a little bit closer and noted that there were significant differences with. For example, eotaxin, some of the CSFs, LIF, LIX, MCP1, and then some of the interleukins. This is just a few of them. There are a few others that do have significant differences. But mainly what's of interest here is that we have cytokines that are important for stimulating the hematopoietic lines and for creating a pro-inflammatory environment. Okay, so I'm going to switch a little bit back to the adipose tissue. Because of course we're primarily interested in how obesity and how increase in adipose tissue can affect our pancreatic progression. So I mentioned that we imaged our mice prior to harvest. We looked specifically using CT imaging so we could image the amount of adipose tissue gain during this time. You can see that our wild-type mice and our KRAS mice both gained adipose tissue on this diet. We basically took a little slice out of the abdomen and used a household unit parameter to actually quantify how much tissue gain there was. So there was a significant gain in adipose tissue with our wild-type mice and with our KRAS male mice specifically, and this correlated well to weight gain. Okay, so we wanted to look a little bit more clearly on the histological features of our adipose tissue because obesity and inflammation are pretty intimately linked. In general, in the obesity literature, if the adipose tissue is inflamed, there is a clear there is a structure that's defined as a crown-like structure, which is necrotic adipocytes. They're surrounded by macrophages. This has been characterized in the obesity literature. When we looked at our mice on a control diet, you can see there's very little signs of inflammation either in our males or our females, and these are wild-type animals. When these mice were fed the high-fat diet, however, you can see that both in the visceral fat and then the peripancreatic fat. I should also mention that when we looked at our fat specifically, we harvested separate depots of visceral fat. So our visceral fat, which I'm talking about here, is fat from the lower abdomen, whereas our peripancreatic fat is that immediately adjacent to the pancreas. When we looked at the visceral fat of our animals, you can see these crown-like structures in multiple areas of the adipose tissue with our wild-type male mice as well as with the female mice, and then these large lymphoid aggregates in the peripancreatic fat. When we looked at our KRAS animals, we saw very similar results. However, we noted there was a little bit more inflammation at baseline, and looks like it was specifically in the peripancreatic fat. So these animals on the control diet, very little inflammation in the visceral fat, a little bit of inflammation in the peripancreatic fat, and then animals on a high-fat diet, the visceral fat clearly looks inflamed. The peripancreatic fat of both female and male animals is really quite striking. So we obviously had to quantify this data, or these data. So when we looked specifically at the male mice, visceral fat inflammation is quite increased, peripancreatic fat also increased. With the females, we noted that when we quantified the data that the visceral fat didn't seem to be as inflamed as our peripancreatic fat. And these numbers are actually quite similar in our KRAS mice in that there is an increase in visceral fat inflammation, an increase in peripancreatic fat inflammation, though not of significance. And then the female mice, again, not really an increase in visceral fat inflammation, but peripancreatic fat is inflamed. So given this amazing inflammation that we saw, we wanted to, again, characterize what cells were made up in this infiltrate. So this is just a slice of a mouse. We just started doing this characterization. So this is H&E staining of a KRAS female mouse, the peripancreatic fat. And we looked with CD45. You see that this large area is likely inflammatory cell origin, and it's just completely very well stained. We look specifically for T cells and for macrophages, and you can see there's some staining here for their T cells, and there are also macrophages here. There's a lot of area that's not stained, and so we're continuing to kind of characterize what this area is. So with that, I'd like to kind of summarize what we've seen. A diet that's high in fat, high in calories leads to obesity in our pancreatic, or in our KRAS mouse model. We saw metabolic disturbances that are similar to the metabolic disturbances generated with in obesity with humans. And that we also saw that a high-fat diet leads to an acceleration of PanIN progression. We saw changes in cytokine and chemokine levels, and we saw an increased inflammation both in the pancreas and in the visceral adipose tissue and the peripancreatic adipose tissue. So with that, I'd like to thank you for your attention and thank our collaborators and, of course, my lab colleagues. Thanks.